In the respiratory tract as well as in the digestive tract, this is called the lamina propria as well. I didn't emphasize it in our respiratory tract, but you more often see it here. And then to help increase the surface area by contracting, we have a thin layer, 2 to 3 cells thick, of smooth muscle. Okay, so that's right here, and that marks the end of the mucosa. Now, we only see that thin layer of smooth muscle where we have smooth muscle in the wall of our tube. So we don't have it in the mouth, and we don't have it in the upper part of the esophagus where we have only skeletal muscle. Okay, but it's going to be part of our general plan, and as we go through, I'll say whether or not it's present. And this is known as muscularis mucosa. Okay. Then just deep to that, we have the submucosa. This binds the epithelium to the deeper muscle layer. Right? So that kind of shows the tube, but the layers are rather thin. So this might show it a little bit deeper. So here's the epithelium, here's the loose irregular connective tissue, here's the muscularis mucosa. All of that is mucosa. This is the submucosa. Right? That's dense irregular connective tissue. And with it, larger blood vessels, larger bundles of nerves, larger lymphatics. This is also showing a lymphatic nodule. All right, so in the ileum where we have the Peyer's patches, that's where all those lymphatic follicles would be that form the Peyer's patches. We also have submucosal glands to help produce additional mucus in some locations. And again, we'll talk about that as we work through it. Oh, I forgot to add my other epithelium here. Sorry. From the stomach to the rectum, our epithelium is simple columnar. Would we have cilia or microvilli? Microvilli. And most of the time we do have goblet cells. Not in the stomach, but in the rest of the tract. So what would be the purpose of, now the respiratory tract, we had goblet cells secreting mucus, and the cilia moved the mucus. What was the purpose of the mucus? Trap. Be sticky? Yeah, dust particles and dirt and particulate matter. We didn't want moving deeper into the airway. Are we worried about dirt in the digestive tract? Are we trying to trap dirt? No. So what would be the purpose of the mucus there? Even in the stomach, where we don't have goblet cells, we have mucus glands. Protection against what?
Bacteria and stuff.
No. Stomach acid, digestive enzymes. Everything that we're breaking down, you guys that eat meat, the enzymes are breaking down the meat in your digestive tract. That's what your body's made up of. Same kinds of cells. So if we didn't have that protective mucus, the enzymes and the acidic pH or the alkaline pH would eat holes like ulcers in the lining of your cavity. So the mucus is there to protect the cell. They undergo reproduction, cell division every 1 to 3 days and replace themselves pretty rapidly. But another form of protection is this layer of mucus. Okay? All right, so the submucosa is going to have everything that your regular tissue has, larger blood vessels, larger nerves, and it binds our mucus layer, our mucosal layer rather, to the muscle. The muscle is what creates the motility, right? It's what moves the food through the digestive tract. So this is known as the muscularis externa, to differentiate it from muscularis mucosa. Our typical plan, our typical orientation is the 20 feet of small intestine, where we have two layers. Okay? There's an inner circular layer and an outer longitudinal layer. And the stomach, because the stomach twists, we add a third layer. In the large intestine, we lose most of the longitudinal layer. So again, we're going to make changes. But our typical plan is an inner circular and an outer longitudinal. Smooth muscle. Okay? So the inner circular is in a circle around the tube, and the outer longitudinal runs the length of the tube. This allows for peristalsis. The same function moves urine through the ureter. It doesn't just fall to the bladder. It's moved via peristalsis. So we have a faculty member here. She hasn't taught 431 in a few years. She teaches the non-majors biologies now. But when she taught 431, she would illustrate peristalsis by bringing in her pet rattlesnake. One day I walked into class and there was a little white Chinese takeout box that was kind of scrabbling and shifting around on the table, and I looked inside and it was a little mouse. And I said, "What's this for?" She said, "I'm bringing my snake in later to show them peristalsis." I thought, well, I'm not going to have a snake in my office, I'm sorry. But the way I illustrate it is just to ask you to visualize, you guys have all lost a drawstring at some time or another where it's come out of your pants or your sweatshirt or whatever, and how do you put it back? Get a great big safety pin, put it through the end, close it first. You never done this before? Okay, this is how you do it, Corey. Take the safety pin, find one of the openings that it's supposed to go through in the loop, push it in, and then you hold one end of the safety pin through the fabric and push the rest of it onto the safety pin, grab it this end and pull it out behind. And keep repeating. Pushing the fabric onto the safety pin, grabbing it in front, pulling it out behind. Grabbing onto the safety pin is the function of the inner circular layer. The outer longitudinal layer is how it shortens and then lengthens behind the food. And so that progressive movement of the contraction of the circular and longitudinal is how the food moves through peristalsis. Okay? The extra layer we add in the stomach allows for the churning that we'll talk about on Monday. And then finally, our outermost layer, the surface layer, out here, is our visceral peritoneum. As part of our 4 layers, it's known as serosa. But you may call it visceral peritoneum. Either term is acceptable, kind of like epicardium and serous pericardium. I'm trying to say right peritoneum and say pericardium, it's not working. So this is attached via loose, irregular connective tissue and secretes a watery serous fluid via the simple squamous epithelium. Okay? Now, any organ that is intraperitoneal is going to have a serosa. What about our esophagus? Is that going to have a serosa? Or it's in the thoracic cavity? Is there any peritoneum in the thoracic cavity? No. Pleura is associated with the lungs, and pericardium is associated with the heart. So the esophagus, as it passes through the lungs, just has dense irregular connective tissue. Okay? So a tunica adventitia, like we had on our outer layer of blood vessels, until it passes into the abdominal cavity, and then it takes up a serosa. So that's what I mean by looking at changes, alterations. This is our basic pattern. And then as we go through the tube and talk about each component of the digestive tract, we'll look at variations. But this is the overall pattern. All right, so we're going to finish up. This is right where we were this morning. It'll be about 10 more minutes. Okay? So I was going to start talking about, I know you just really made me feel good about letting you hear the sound of my voice. So I was going to move into the mouth, but since we're going to combine anatomy and physiology, I want to do an intro. This is kind of our anatomy baseline. But let's flip over to page 42, I think, and let's look at the intro to digestion. Try to lay a framework for that. If you had nutrition, this is really, really basic, okay?
Do a good job, it's been a while.
Okay. All right, so remember at the very beginning, when we were talking about, an hour ago, when we were talking about the mechanical and chemical digestion, hydrolysis is another term for digestion. And it works both ways. We use water to split our chemical bonds, and water itself is split. And I actually, when I first started teaching, I had a video, or rented a video, got a video from the library, where they took a sterile piece of meat, a small piece of meat, and put it in a sterile jar of water, in a sterile string, and then took video of it and speeded it up, and you could see how it got. Pieces would fall off, it would break down. Because all you really need for digestion is water. All right, we have two carbon bonds. A carbon bond, add water, and it will split and break the carbon bonds or the carbon-hydrogen bonds. But it took like 2 weeks, 10 days for this to happen. It was a small piece of meat. So to increase the rate of the reaction, our body synthesizes enzymes that are added to the lumen of the digestive tract to increase this rate. Now enzymes are very specific. Okay? If we have a protein with a sequence of amino acids, there are certain enzymes that will break apart the amine side of the protein and release amino acids and only work on this end, called aminopeptidases that we'll talk about on Tuesday, and other chemicals, enzymes that will only break down the acid end, okay, carboxypeptidases. So they won't work in the middle here. All right, so we have these reactions have to be governed in a particular sequence. We have three basic food types: carbohydrates, proteins, and lipids, or fats. For carbohydrates and proteins, the digestion is done in 2 steps. An intermediate digestion. This is done with a common enzyme by the name of amylase. As we'll see on Tuesday, the salivary gland, the parotid gland, makes amylase. You probably didn't, you've probably been told, but I doubt most of you do it. If you take a plain cracker or a saltine cracker that you've brushed the salt off of and chew it for 5 to 8 minutes, it will start to taste sweet. All right, that's the salivary amylase breaking down the starch of the cracker into a disaccharide. Sucrose is one of our table sugars, one of our common disaccharides. But that's too large to cross any of the membranes in our body and be taken up by our body, all right? So it's not going to be able to, it's not available to stop digestion in your mouth. That is not available to your body for energy. We have to do another digestion with enzymes made by the small intestine to break that. These will not work. Maltase will not work, or sucrase will only work on sucrose, a disaccharide. It will not work on starches. So if we only have starches, that won't work. This is why someone who has lost their pancreas, the major source of amylase, through pancreatitis or pancreatic cancer, cannot eat normal food. I had a student at Sac State whose uncle had gallbladder stones that blocked off his pancreas, and the pancreatic enzymes and bile backed up, not bile, but bicarbonate ions backed up, And 2/3 of his pancreas had been digested as if it was food he was eating. And he had to have the rest of it removed. And so to eat, he had to have a line, a large needle into his subclavian vein, and he wore a backpack with a pump, and it pumped in pre-digested nutrients into his blood. Because he had a small intestine making these enzymes, but he had no pancreas making this enzyme to create the substrate for his small intestine. So it wasn't able to work on it. Okay, so that intermediate stage has to come first, and then finally the final stage provides the individual sugars that can be absorbed. So as we go through, we'll talk about the enzymes that work at each unit and the type of digestion that's taking place. But I wanted to give you this precursor to it. Proteins are very similar. We start out with large proteins, and we have an intermediate stage. All right? So in the stomach, we have pepsin, and the pancreas makes trypsin. But it won't completely break it down. It'll break it down into 3 to 5 or 2 amino acid sequences. But here's the aminopeptidase and carboxypeptidase. Well, finally, the amino acids is the smallest thing that can be absorbed. So again, without the pancreas, we can't break the proteins down into individual amino acids, which is the form they have to be in to be absorbed across the body wall. Finally, this is the last slide, we have lipid digestion, which is interesting because lipids take a long time to digest, but there's only one step. And we're looking at triglyceride here. Obviously, there's cholesterol and other forms of lipids. But triglyceride is formed of a 3-carbon molecule, a glycerol, and 3 fatty acids. The enzyme lipase breaks off 1 or 2 of these fatty acids, and the molecule can move across the plasma membrane of the cell wall. We're going to be doing this actually on Thursday of next week. We're using a pancreatic mix that has lipase. I bring in whipping cream, and the lab tech mixes it with a purple powder called litmus powder. And once digestion has started, it turns pink. That's how you know alpha releases a fatty acid that stinks like sulfur and rotten eggs. A little bit. It's not going to stink up the room. Why do you think it turns pink? One of the questions on your lab report. What's being released that's going to turn it pink? You guys are tired. The answer's on the screen. Fatty acids. Okay. So as the lipase starts to release fatty acids, that is going to lower the pH, and it's going to change the color of that litmus powder from purple to pink. And that's how you know that digestion is occurring. Okay? We'll stop there. On Tuesday when you come back, we'll start with the mouth and go through each of the components of digestion and how they're organized and what happens there. It's secreted in the mouth, and most of it in the pancreas.
She's got to go. She's going to When I study it, that's one thing that got really bad.
Only ways that mom can. And when I said it, I was really just like,
Is it just a few?
oh, it's okay.
It was really cold.
Okay, this is stuff they got her oh yeah. I think it's very jealous. She went to she needs to do 2 separate orientations.
Yeah, and I'm just like, sometimes look at baby run.
Yes, we did. Oh, there's another one? Yeah. We came and did a tour of the Canada kidney and everything, so I mean, they're all different. You can't share it's different.
Yeah, exactly. Oh, he does it, especially when I'm showing the little one.
One of the drops, did you ever get a strawberry? Yes. And they have like, okay, the one that drop, yeah, she's not here anymore. If you want to see somebody else, okay, it might be a little late if you guys are reaching down for. Yeah, it's okay. Oh. Well, then just by the time that I get it right home, like, yeah, I really want to go. Where's your clinic at? Huh? Where's your clinic at?
And that's for the actual college, not for a program.
So you,
Yes. I love these shirts. I like them. Yeah, they're really soft, they're really cheap, and I love it about everything.
Is it still like me next? Karen and me both came in.
That's funny. I didn't know that was the session. There's a boyfriend tee on the tab. There's a boyfriend tee on the tab, yeah. I don't want to.
So let's say she's not white, and
No,
and they still have the orientation for sex.
Is it to go to office?
Okay.
And how many coupons, like, can you just make her limit?
How many is your left go? How many can you buy with 1 coupon? Like 3 to a coupon?
So I will totally do that. I'm gonna do that tonight.
Probably gonna
So
take
it's
towards.
to win?
Okay, so how do we sign up?
You guys are very fine.
On how to sign up, and then there was some type of a fee that's required that you were gonna try to get.
Okay, good for you. I just not really good. I don't know what's happening. Last night, my follower, no, doesn't have their names actually like down the line. It was originally here, and it's still there, but it's like going down my leg now. The front, just the front, and like, and then it might, and I was like online, so. And then that was on the test day, and I got that wrong. I was looking at this for like 3 hours last night. I went to Quadriatus Lumbar.
Yeah, that was the narrow one.
Yes, I was like, but I was thinking more of the front leg.
Do you know
It
what
goes
the
just to
fee is
the,
for
let's
her?
try to enter, just to go beyond that.
Correct.
Yeah, so I was like trying to figure out.
Okay, and then that can be
Nerve goes down there, and it has branches that go out the side.
So like.
Has to be
But it-
when she signs up.
Sometimes it can get pinched.
Yes.
Where it goes over the pelvis,
And so
but if it's more numbness than
I don't
pain.
know, I don't know, like, I'm trying to figure out what, because it's like.
That's what's for you, she kind of moves around.
So, but.
Okay, and then if,
but now it's
we're not able to.
What about your chest?
If we're not, if she's not able to find it online, is there a number we can call to sign up, or we have to do it online? That's the only way.
Yeah, rubber balance.
Okay, can we do that? And